Hello everyone. Today I'm going to discuss about DNA replication, an important part of the CSIR exams and other exams. Now when I talk about DNA replication, first of all we should know what DNA exactly is. DNA stands for deoxyribonucleic acid and it is a double stranded structure. Okay. When I say DNA replication, here I mean is the exact mechanism by which DNA is copying itself or DNA is making its copies. Okay. So inside the body, inside the cell, what is happening is and during this DNA replication process, first of all, since DNA is a double stranded structure, so there has to be a separation of these two strands. This process takes place with an enzyme called helicase. Now this is separating the two strands, or you can say it is unzipping the two DNA strands. Now we have a single strands. Okay, now what happens is your DNA polymerase would come into an action, and it will add complementary base pairing amongst the strands. Okay, now this process, during this process, one is your leading strand while other is your lagging strand. Okay, so when I'm saying leading strand, so in leading strand continuous replication take place, while in lagging strand continuous replication doesn't take place. Instead there is a replication in the fragments, and those fragments are known as your Okazaki fragments. These Okazaki fragments are later on being joined by DNA ligase. Okay, so three enzymes are playing important role over here. First is helicase, then your DNA polymerase, and then your helicase. Now scientists have mimicked this basic technique, basic principle of this copying of DNA, and they have been amplifying a lot of single specific DNA into millions of DNA, or making millions of copies of DNA, and that process or that technique is known as your PCR, which is known as polymerase chain reaction. Okay, so now what happens in PCR? The process basically means making copies of DNA. In PCR, there are three steps. One is denaturation, then your annealing, and then extension. In the process is almost similar. Here, what happens in a denaturation? What is happening? Your strands are being separated. But here, instead of helicase, here heat is being used. Okay, so the denaturation takes place, followed by your annealing, where we are using oligonucleotide. They come and bind to, they come and lead to formation, they come and bind to the specific area where for which they have been designed. Okay, so the basic specific sequence which has to be extended, that is being targeted by those primers. Then there is an annealing, where DNA Taq polymerase come into action, and it again do the complementary base pairing. Now here Taq polymerase is used instead of self DNA polymerase, because Taq polymerase is your heat stable. So now from 1 copy of DNA, for one specific target DNA, millions of copies is being produced, and there is an amplification of millions of DNA. This process, this technique is known as your polymerase chain reaction. Now this polymerase chain reaction has been used extensively in various laboratories for various research processes. One of the example is like once you have isolated the DNA, and then you did the PCR following that denaturation, then annealing and extension process. Now you have copies of DNA. Now you can add restriction enzyme to it. Restriction enzyme will cut the DNA at specific part, and then we can study like of what kind of a particular genotypes could associate with a certain kind of a disease. So in also like this particular process could also be helpful in DNA fingerprinting, in diagnostic studies. So this technique has an important role in research. So both DNA replication and this PCR amplification process, they work on complementary base pairing. Okay, so this is what I am going to teach today. Thank you so much.